Have you ever seen bacteria that glows in the dark? Probably not, because it doesn't normally glow in the dark. But this is a special bacteria that I've genetically engineered to be able to glow under a black light. You might be wondering how we can genetically modify bacteria to glow. But first, you need to know about the genetic material within a bacterial cell. To begin, plasmids are circular pieces of DNA that roam free within the cytosol of bacteria. They carry DNA a lot like the chromosomes that we see in the structure of our very own cells, except that these plasmids are not part of the genomic DNA, and they can be easily taken out and altered. So how do I alter plasmid DNA? Well, it's actually quite simple.
The plasmids are cut open inside of the bacterial cells using restriction enzymes such as HIND3. In its place, the gene for green fluorescent proteins is added in, which make the plasmids start producing green fluorescent proteins. And now the E. coli glows.
Now that the plasmids have been altered, let's move back into the lab so that we can prepare the E. coli. And the next step is to take your host cells with the bacteria and place them inside the solution with the CaCl. And this increases competency of the cells and basically opens up the lipid bilayer to allow foreign DNA to move in and be absorbed by the bacteria. In tandem with the calcium chloride solution, we also use heat shock, which involves first putting the solution into an ice bath and letting it rest for 10 minutes, which expands the cell and increases the cell membrane. And then, immediately, the solution is taken out and placed into a floater to be put into a heat bath, which will rapidly shrink the cell back to its normal size. After repeatedly doing this for 2 to 3 times, immediately following each step, the cell membrane will weaken and the foreign DNA will be able to be taken up into the cell. And then finally, the plasmids with the gene of interest will be taken out like so, and then placed into the solution with the weakened host bacterial cells to hopefully be taken up for transformation. This next stage is very important as the cells will need to rest before they can begin to grow exponentially. So, a recovery solution will be added to the solution with the host bacterial cells and the plasmids with the gene of interest so that the cells may rest and get the nutrients that they need to grow exponentially. The cells will rest in this recovery solution typically overnight until they will be plated onto an agar plate like this, and then hopefully grow exponentially and glow. And before you know it, you've altered the DNA of bacteria. Afterwards, we remove the glow-in-the-dark protein, and we can use it as a fluorescent tracker to observe how cancer and other diseases move around within the human body. Tons of other products such as artificial insulin and artificial human growth hormone have been made using the same technique, which have saved millions of lives. And there's still so much more that can be done.